Hello A-level biology lovers, my name is Dukunimana Paul. Today is episode number 3, unit 3 called microscopy. In video number 1 we have talked about introduction to biodiversity. In video number 2 we have talked introduction to classification. Now today video it's all about microscopy. So we have started the journey of describing and explaining biology deeply from senior 4, senior 5 up to senior 6. What I'm requesting the students who are understanding me is just to make subscribe on this channel, comment and share so that their grade in biology should be A positive. So it means that you have realized that many students they are suffering to get good grade in biology. So I hope the student who choose my channel he's in a good way, so he or she can expect good marks in the biology. So today's video it's all about microscopy and they are relevant. So let's continue by talking about the table of content. So the table of content today, their details, all of them, they are in microscopy. We are going to cover everything on microscopy. So sit down and have pen and a notebook, then follow me. The learning objective today is not quite many. By the end of this unity, the students should be able to firstly describe the features and function of the compound light microscopy. As you are seeing here, this is called compound microscopy. They should demonstrate perseverance in using light microscopies, manipulate a compound light microscopy for prepared slides, ensure care to avoid the damaging lens, mirror, and slide of microscopy. You will have the time to understand what is lens, what is mirror, what is slides. So, and also you should be able to define magnification, very very important, your national examination, and also to explain resolution. Also it is very very important in your national exam. So and also they are supposed also to understand the appreciate the importance of magnifying instrument in the biology, which is microscopy. At the end also, we should be able to determine the relationship between actual size and the image size. This here we will talk about the formula regarding how to calculate magnification. Calculate the size of the biology structure using appropriate unity. Identify advantages and disadvantages of electronic microscopies, and understand the principles and limitation of transmission electronic microscopy and scanning electronic microscopy. Compare light and electronic microscopy effectively, and also acquire the knowledge the role of electronic microscopy in modern science, and also able to observe and draw biological specimen using light microscopy, and also to prepare temporary slides using different objective lens. So I hope you are understanding. So today you have the learning objective, which is not quite many. So I encourage you to have pen and a notebook and then sit down, then follow. So let we start. So unit number 3, which is microscopy, the K unity competence, it's all about to distinguish between types of microscopies and their uses. Means we have only 2 types of microscopy. Type number 1 are light microscopy, type number 2 are electronic microscopy, and all of them are subdivided. So I'm encouraging you to sit with me, understanding me, raise the volume, study hard. Success is not accident. I hope you are understanding. Thank you. So let me start by defining what you call microscopy. What is microscopy? What is microscopy? So microscopy is a technical field or a technical instrument for viewing object not visible to the naked eye. Means are the instrument which we use to see the object which is not visible by using our naked eyes. Means I hope you are understanding. When we say micro, it is a small. Scopy to see means are the instrument used to see the small microorganisms. I hope you are understanding. That means once you I remember in the unit 2, I have taught you about the introduction to classification. We have understood the some unicellular organism. Those are unicellular organism like plasmodium, those amoebas, protista, all of them, monera. The organism in the kingdom monera, you cannot see under naked eyes. You are going to see kingdom monera and some of protista, you're going to see them under microscope. So that's why microscope, it is very, very important to see very small organisms. So by continuing, I cannot continue without talking about you who discover microscope. And once I see there's someone who discover microscope, you should clap for him or her, because he did a very great job, because they are the one who discovered everything you are seeing. So number 1 is called Zacharias Janssen. So this one is the one who created first microscope. Means it was in the 16th century. I hope everyone is clapping for this man Zacharias Janssen in his or her heart, because he's the one to create first microscope. So even if we create the first microscopy, this microscopy is not light microscopy and is not electronic microscopy. If they ask you who developed or who created light microscopy, you should answer Anton van Leeuwenhoek. So this is the man who called Anton van Leeuwenhoek. This man is the man who created. To read it is not easy, but you should pause and review. Once you get your multiple choice question, this is the man who created the first light microscope. I hope you are understanding. It is called Anton van Leeuwenhoek. I hope I tried. So means like he did this in the 17th century by seeing the cells. So after this man, Anton Leeuwenhoek, who discovered and developed light microscopy, also later on, like 1930, also electronic microscopy also was invented to allowing observing the organelles. Organelles, you remember, in the cells, without using light microscopy, you cannot see anything. So when you see by using light microscopy, cell organelle, we are going to see only just 3: cell membrane, cytoplasm, and nucleus. Those mitochondria, ribosome, and so on and so forth, we use only electronic microscopy. Means under light microscopy, we can only see few organelles like cell membrane, cytoplasm, nucleus. But by help of electronic microscopy, you can allow to see now more organelles like mitochondria, chloroplast, smooth endoplasmic reticulum, and so on and so forth. And then you should make subscribe, because the next video, it is unit number 4 called cells. I hope you have already understood the definition of microscopy as instrument that is used to see a small object you cannot see using our naked eyes. Definition is here. The history is not quite many. Just understand that the man who discovered and developed light microscopy, it is called Anton van Leeuwenhoek. So I hope I have tried. So you should understand that each microscopy, they have the parts. Okay. As the human body also have parts, head, what. Also microscopy, they have head, maybe abdomen, also legs, but the parts are not the same. You should come and here and memorize each part of microscopy. And after this video, I'm telling you surely that you're going to be able to explain this right microscopy. So this is compound microscopy. It is not electronic. This one is right microscopy. So features and functions of microscopies. So first of all, the main features. Each microscopy, they have what you call lens. Do you know people who are wearing glasses? That glasses, they contain lens, which magnify or increase the size of maybe object that people who suffering the eyes, they are looking. So means also in microscopy, there is lens. Lens there, it is there here in A called the ocular, and the same case also lens are here in objective. I will teach you this. But each microscopy should have lens. The function of the lens is just to make magnification, to increase the size of that small object. So other things you have to keep in head, light microscopy, it should have also light source for illumination. So it means this M, it is mirror or light source, means that by using this M, it will put light via to the diaphragma condenser. Now the light will be there in this light so that you can be able to visualize your specimen. Specimen are those sample we need to increase. Means those objects which is small we want to increase are called specimen. So let we have the look on the functions of microscopy. As we have said, function of microscopy is just to observe a small object which cannot be seen under naked eyes. So means those small to observe it, you are not going just to take stool if you want to see the parasites or bacteria in the stool. We are not going just to come and take a stool and then put here on this stage. No, here means we should have what you call microscopic slide. Means this microscopy, the function of it is to observe prepared slides. Means in this prepared slides, what is prepared on is that sample you want to observe. Means if you want to observe the bacteria in the stool, means just take a stool and then make a preparation of the slide. Then you put the slide contain the stool here. This one you are going to call it the stage. Now here we're going to be able to observe what is inside in the stool. Is bacteria in or not? So means also at the end of this video I will teach you how to prepare microscopic slide. How can you make slide for observing under light microscopy? So other function also is just help to increase to magnify your object. Means you are observing small substance. Means the function of this microscope, it make magnify, it increase the size of your object. Means it increase in apparent size. Okay, and other function of the microscopy also, it's what we call resolution. Resolution is ability to distinguish two separate objects. Means suppose that, let me have the marker. So suppose that this one in the sample we have this one as bacteria. Under when you are viewing under light microscopy, we are going to see this one as like this one, like this. Means it's going to make what you call magnification. This is what you call magnification. Okay, to increase in the size. So what do we call resolution? Means in here is where we put our microscopic slide. Suppose that the slide to be observed is this one. Okay. Now what you did, you come and then you put the sample. Maybe it is stools. Okay. It is stools. Means you have put this slide under this stage. Okay. What are you going to look? Is it the stool containing some? Is this stool having some bacteria? Means like ability of the microscopy. Okay. To distinguish these two objects, this one and this. Okay. To see them separately. That is what you call resolution. This is what you call resolution. Means what is the difference between magnification and resolution? So when we say magnification, it's all about increasing in the size. When we say resolution, it's all about ability of microscopy to separate 2 points, okay, which is being observed. Means 1 point here, another point here. Means if the microscopy, they have high resolution power, you will see one bacteria here. Means they will put space. Now put another bacteria. Means here you are able freely to observe your bacteria separately very well and then study them correctly without making the mistake. But imagine one bacteria is here and then another bacteria is here, are congested. How can you separate these two bacteria? Means this microscope is not good. They have low resolution power, this one, while this one they have high resolution power because it is able to make a space, ability to distinguish two separate objects. So this question, it is asked in your national examination. So I'm advising you to take in your head what is magnification and what is resolution. When we say magnification, you have to answer is increase in apparent size, while resolution is ability to distinguish two separate objects. I hope you are understanding the keyword, which is very important in this video unit number 3 called microscopy. Thank you. So let me have look. Means I told you in the beginning that usually microscopy, they can be either light microscopy or they can be either electronic microscopy. Those are two types of microscopy. They can be either light microscopy or electronic microscopy. So let me start in number 1 called light microscopy. So when we say light microscopy, you are understanding word light. Okay. We are saying light microscopy. Okay. Means this one we are going to talk about light microscopy. So by definition, light microscopy or compound light microscopy means it is microscopy that use light to see the object. Okay, it is optical microscope that use visible light and the lens, as I told you, for magnification small object. I hope you are understanding. So this what you are looking here. This is light microscopy. They use light from here mirror, and they use the lens. Lens, usually we have lens here in eyepiece, and then also we have lens in this also it is called objective lens. Means by help of this light from mirror, means the function of mirror is to produce light, and now by help of the magnification which found in this lens in the eyepiece, and also the lens in the objective lens, we are going to have our object which is being magnified. So I hope you are understanding what is compound light microscopy. So let me repeat the definition of light microscope. It is optical microscopy or is microscopy which use light and lens to magnify small sample. I hope you are understanding. It means you are having stool, you want to observe the maybe stools, bacteria. You're going to prepare slide, as I told you. Means by having the slide, you will put here on this stage. Now you will observe here, you put eyes here on the eyepiece. Then you are going to observe what you call what is inside in your slide. Okay. Like it is like this one. So when you look it very well, it's like, it means you put slide here. Okay. This is where you put the slides. Now your eyes, human being, are here now observing what is inside on the slide. So this is what you call compound microscopy. Okay. Other things also means the compound microscopy. The compound, there are types of compound light microscopy. There is what you call monoocular light microscopy and biocular light microscopy. When we say ocular, when we see the part of microscopy, I will tell you that the ocular is this one. This is what you call ocular. So this ocular, if it is one, it is called the monoocular. There is some microscopy which have more than one ocular. Means this microscopy which is look like this. This one is called the ocular. While if we have only one, like this one is called the monoocular. So means by coming here, I hope you are able to understand that types of microscopy are either monoocular or binocular. When we say monocular, means like this one. This one is monocular. We are having only one ocular. Ocular, it is also called the eyepiece. Means only one eyepiece. This microscope, it is going to be called the monocular. When we have two eyepiece, it's going to be binocular. Means when we say monocular light microscopy, they have single eyepiece. Okay. Now when we say binocular, means B is two. Binomial nomenclature. Do you remember binomial nomenclature? Just to give organism two Latin names. So B, it is always coming. Monocotyledons. Okay, one cotyledon is like a maze. Okay. So I hope you are understanding the difference between two types of compound light microscopy. But in your national examination, please understand the two words here I told you, magnification and resolution, because microscopy are going to use the two so that you can see the image of your object. I hope you are understanding. So after understanding this light microscopy, let's continue by understanding their main parts. I hope everyone supposed to sit down, take notes, sit down, then I have to teach you how to name the parts of the compound microscopy. So I'm going to do it in this way. Okay, are you ready? Are you ready? So this is a microscope. When we say base, let me show you base. Means base is this one. Okay, on microscopy it is stabilized, means if they ask you the function of the base, okay, if they ask you the function of the base, just come and say, sir, function of base on microscopy, it's like ichibuno, okay, means it is base. It help the microscopy maybe to be stable on the table or any ground it is sitting. Okay, means the function of this base, it is called to stabilize the microscopy. So if we come here on light source, it provides illumination. Means light source, it provides light. Illumination, okay, means remember light microscopy, they use light and lens so that we can get the magnifying the image. Okay, this one, it is called light source. When we come here on the stage, okay, stage, you understanding stage, where we put things, okay, means on the stage is where we put our slide which contain the sample which has maybe bacteria, means we just make like this. Okay, suppose that this is a slide. Okay, in our slide we will put sample, maybe stools or any, we are suspecting bacteria in this stool, we will put here in this called stage. This is what you call stage. Now this stage, their function is to hold the slide in position. Means once we put our slide here on the stage, it will keep the slide in position so that people, they can come and see it. I hope you are understanding. So when we come here on the part of the stage clip is just to secure the slide on the stage. Once you put your slide on a light microscope, you should place things like this. You should place it in the position so that it cannot move. See this one, you are looking this one, means after putting your slide, you will just come put something which can hold and then secure in the position. Okay. So by continuing arm, okay, when we say arm, it is here. Okay, this is arm, means when you're going to take microscopy, you will take your finger like this. Okay, remember to take microscopy with two arms, not single arms. So this G is called the arm on microscopy. Their function is to support the body tube. Okay, body tube I will show you, and also is used for carrying. When you are carrying microscopy with your hands, it means their name, it is called the arm. Okay, is where arm your arm is going to hold. Okay, means this is the arm, it holds what you call the body tubes. When we say body tubes, are this one G. See this G, it is held by this G. Okay. Now when we come here on what we call coarse adjustment, okay, I told you that in the microscopy we are having this A, where we have the lens and this objective lens. Okay, means sometimes we want to make this objective lens down or up. Okay, so that they can come closer with the slide we have put it here. Okay, means if we want to see image in this slide very well, we will move down here this objective so that the lens can see it very well clearly. Okay, means this adjustment means they are going to adjust the distance between the slide and the objective lens, which is this one. Okay. Now let me talk about what you call cos focus adjustment. Means they do for large movement to focus. Okay. Means when we say cos focus adjustment, their function is for large movement to focus. Means once you move, you want to move those objective rapidly, you will use this B, which is cos focus adjustment. So when we say fine, it is small. Okay. It's just for precise. Means you already near the slide. Okay. You are looking the field. Okay. Means after, what is the function of the fine focus adjustment? It's just for precise focusing. Means you just precise where exactly there is your image bacteria or your purpose you are identifying in your sample. So I hope I tried my best. Base is for support microscopy. Right source is just provide light. Stage is to hold the slide. Okay. Stage creep is to keep the slide in position. Arm is for support and transport. Cos adjustment, it is just large movement to focus. Fine adjustment, it's just precisely focusing. I hope you are understanding this function of the microscopy, and then you are able just to names. Okay. Now let me easy life. Now here I have prepared for you. Base, it is R. Okay. Now M, it is this one. Stage I. Okay. Stage is I which is here. Okay. Now stage creep, it is J. J is this one. Okay, remember this one. Okay. Now arm is G. Remember. Now cos adjustment, it is B. Then D, it is fine focus adjustment. I hope I tried my best. Let me continue with other remaining parts. Okay. Now I hope you have tried. I told you that this one is base. Okay. This one is mirror or light source. Okay. I teach you this one. It is cos adjustment. This is fine adjustment. Okay. This is arm. This is stage. Okay. Now this is the stage creep. I hope I tried my best. Also their function you should know. Pause and review. This is for support, right source, precise refocusing, moving objective rapidly. This one is where to put the slide. This one they make the slide in position. Okay, let me understand the other parts. Other parts, it is just for optical component. Means their function. Means how to increase, how to make magnification. I told you that there is what you call eyepiece and objective lens. Means we have what you call eyepiece, also called the ocular lens. Okay, and also objective lens. Means A is the one which is called the eyepiece. Okay, let me just join them. Okay. Let me join them like this. Okay, means objective lens are this one. Okay, or FNHR or objective lens. Now eyepiece, it is here on A. Okay. Now revolving nose. Okay. Remember, nose is just to remove the mucus. Now here also this one nose, it's just to switch or to move between the different objective lens. Remember, the objective lens, you have 3 objective lens. Okay, all of them are not the same. Maybe this one it is 100 for magnification. Okay, maybe this one is 40 for magnification. Maybe this one is 10. Suppose that you want to increase the size of your image you are looking, so you should use this revolving nose piece just to switch or to move or to interchange the objective lens. That is the function of the revolving nose piece is to switch between different objective lens. So when we come here on means this one objective lens and the eyepiece, their function, all of them they are the same. All of them are magnifying, is to magnify specimen. Means you need to increase the size of what you are looking for. Okay. So let me come on the things which are responsible for light management. So here this K, usually it is called diaphragma. Okay. Diaphragma means light come and now they hit on the mirror, it reflects it automatically here in this diaphragm. Now the diaphragm will control amount of the light which will come and pass through the slide where the specimen are. Okay. When we say now the diaphragm, it regulates amount of the light intensity for viewing our specimen. I hope you are understanding. Now when we come to the condenser, it focuses the light onto the specimen. Okay. Like if this one is the slide, okay. This is the slide where there is a sample, okay. Means the part of microscope which control amount, how many amount of the light which is going to come to pass here in this slide, okay. This is what you call diaphragm, iris diaphragm. Now the part which will focus means the diaphragm, it control amount of the light which will come, how much amount will come. Now when we say condenser, it now just take this light, okay. So what is happening? Okay. When we come here on this diaphragm, it take this light, it condense the light, okay. Condenser, it focus, it condense the light onto the specimen. It take this light, okay. Let me have the marker. So it is not working. Let me use another way, okay. It will take this light and then focus them in the slide. I hope I have tried my best. Let me continue by showing the letters, okay. So it's okay. If you are looking very well, so this one is the lens. It is FNH, okay. Noise piece, as I told you, it is E. Their function is to switch or to interchange objective lens, means to move, okay. Change from 100 to 40 or what. Now ocular, they have the lens for magnification. Diaphragm, it's just control amount of the light which will come to the specimen. Now condenser, it will focus right onto the specimen. Thank you so much. So this is the overview of what I told you. Take time, draw with your pen, okay. You can draw. I'm telling you truth, you can draw. Don't be scared, okay. Don't be scared. You can do it. You can. It is not easy. Just make ocular like this, okay. Now make what you call body tube. Those are the body tube, okay. This is body tube. Usually it is the body tube where we are going to put the objective lens, okay. This is objective lens. Just now come and put arm, okay. So here you can put stage, okay, where the slides and the stage clip are going to fit. Now you can continue, okay, by putting what you call coarse adjustment, okay, and you can just put fine adjustment. Now come here, then put the base. Under this base, you can just put the mirror, and then here their condenser, and then their diaphragm, okay. Now you are done, okay. Those are nose piece, those are ocular, okay. I hope you can do it. Don't fear, you are able, you can do everything you want, okay. I hope this is the sketch of microscopy. Take time and review and understand where there is, because in your national examination they can either ask you the function of each part, as I told you. They can ask you the function of each part, not only function, also they can ask you to name A, B, or C. Okay, thank you. I hope you have understood the main parts of microscope. So let me talk about the basic microscopy care. Means how can you care light microscopy? Means like you take your phone, you just take like you put in where you want. Means you don't care. Means is it the same to take care of microscopy? No, because microscopy, it is expensive material. It should have a specific care. Okay. Like those Rwandan people who still have culture, when they're going to greet elder people, they give 2 arms. That is the same case you should carry your microscope. Okay. You should carry your microscopy with 2 hands. Okay. I hope you are understanding the way. Now, once you're going to keep, means proper to care care number one of microscopy is just you should care by taking two-handed carry. You should carry microscopy with two hands. Other things, after finishing using microscopy, remember to turn it the objective lens two row objective lens. I told you microscopy they have many objective lens. Large, small. Remember, when you finish, just put small objective lens on this stage. Okay. Now other things, safe placement. You should put in safe place. You should put in clear work, means you should clear workplace, means remove unnecessary items from the table. Means once you put microscope here, remember just to remove other things else. Okay. You remember to clean the lens. Okay, there is what you call just clean lens only using paper lens. Don't use your gloves, please. Just there is a paper lens which is only specifically for microscopy. Okay, avoid the towel or tissue to prevent the scratches. Okay. Now other things you should remember, once your microscopy is not working properly, it have a problem. Just automatically report your issue. Just means maybe you are lab technician, or you are operator, you are using a microscopy. Remember, if you are a student, notify to your student. Okay, so notify to your teacher that your microscopy is not working properly. Okay, now you should take responsibility. Treat the microscopy with care. You are responsible for its condition. I hope all of those are the ways. Okay. Not only microscopy and other thing else you think it is very valuable to you, just take it with two hands. Okay. Once you are finishing to use the right microscopy, take it to lower objective lens. Means return to lower power. This one, okay. Objective lens, you should put to lower objective lens. Now keep microscopy in a safe place where there is no robbers. Robbers, they can take it away. Then you are going to feel the absence of it. Okay. The clear place, you should remove all other items and also clean the lens. This is the lens. In this objective, there is lens. You should just come and clean regularly, so that once you clean, you are going to visualize your image clearly. So, and then remember to use only lens paper. Those paper who are only just designed to clean microscope, not your things. You are just paper or other things. So report issue and then take it as your responsibility. So let we come here in the magnification. Magnification, as I told you, it is increasing how much your image is increased. Okay. Magnification is enlargement of the object which is being studied. Okay. Magnification, I have just discussed on the beginning slide as the increase of the image. Okay, magnification, it is enlargement of the object being studied. Okay, it is increasing of the image. Okay, how image is increased means that is magnification. Magnification, take in mind that is magnification, enlargement or increase of the object being studied. So means you can magnify. Once I'm telling you magnification, how much is times? You can make your image, your object 1,500 times. Okay. Means you can take, see this point, you can times this under 1,500. Okay. Like means you can take this under microscope, you can see it like a cow. Okay. If it is like this, you can see under microscope bigger than a cow. Okay. So how to measure magnification? It is easy. Just take actual image, means the image you are looking in your microscopy, okay, and then divide the object, actual size, their normal size. Image in microscopy, divide the actual size, how are looking in a normal condition, okay. So after understanding this, I'm requesting you, before calculating the magnification, make sure that your image and actual size, they are in same unity. Specifically, the unity are supposed to be in micrometer or nanometer. Means all of the two are the unity which are supposed to put actual size and the image, so that you can have your magnification. Means once they give you the unity in millimeter, you have to times 1,000 to convert in micrometer. So let me help you by helping you doing example. You will see this example in national examination. So calculate magnification. If actual size is, this is the normal size, okay? Means under microscopy, the measured image, it is now this, okay, you see, it is magnified very big, okay? Now the formula to use, remember, magnification is equal to image over object, measured image over actual size. This one is the image, this is the actual size. But when you automatically divide, you're going to get 0, okay? Because see, this one, the size, it is micrometer, while the image, it is millimeter. You cannot divide. You should convert this millimeter in micrometer. How? Take your micrometer, then times 1000. Now, by that conversion, you're going to have your micrometer. Now, this is the image in micrometer unit. So, now take your micrometer image, okay? Divide by micrometer object. Now, this is magnification, okay? Remember to put the times signs, okay? Remember to put the times signs under. Remember to put the times signs in your. So remember to put the times sign here on the magnification. I hope you are understanding. So let me talk about resolution. I told this one at the beginning. Resolution, it is ability of microscopy to distinguish two separately objects. So here resolution is ability to distinguish 2 points as separate image. I hope I told you, even I tried to draw for you, okay? I told you if this one this is object, means the image is like this, okay? This is what we call magnification. It means they have increased. But once you are saying, means this is one thing, this is another thing. So this is the image I'm drawing, another image, okay? So this one, it is not all about the magnification, okay? But see, there's a 2 points are not separate as this one, okay? Means this microscope which can give you this image, it have high resolution power, but this one they have low resolution power. So means when we say resolution is ability to distinguish 2 points as a separate image. I hope you are understanding. Means the importance of this resolution, okay? It will help you. Means high magnification without good resolution, your image is going to be vague. Your image is not going to be clear. Imagine that your image has been, your image, this is the image. Means under light microscopy, people who is seeing in this microscopy, this is how in the eyes, in his or her eyes is going to be appearing. This is how the image is appearing in the person who are looking inside the microscope. So if you didn't see the microscopy even at once, suppose that you put eyes here in this ocular or eyepiece, means in your eyes, this is the image which is going to come, okay? Means in this image, see, this one, I'm seeing the point separately. This is point number 1 and this is point number 2. But imagine, this one are congested. They are congested. Means you are unable to distinguish them, okay? But by using this microscopy, you are able to see the background, what and what. But this one, they are vague. You cannot conclude based on those results. Those results are not good. So means when you have not without good resolution, your image is going to be blurry, vague image. They are not going to be not clear. So resolution limitation, it means short distance between two points can still being seen as separate, okay? Means if the limitation means if it is small, this is the limitation. I hope you are understanding. So what are the key takeaway in magnification and resolution? Because by understanding this magnification and resolution, believe me, this unity, the question are going to come from magnification and resolution, either for calculating magnification, either to distinguish them in terms of definition. Say magnification is how much image is increased, while resolution is how image are separately from each other. Okay, I hope you are understanding. Pause and review and understanding the difference between resolution of microscopy and magnification. I hope you are understanding. So now, after understanding light microscopy, it's time now to come and on another type of microscopy, which is usually electronic microscopy. Okay, light microscopy, they use light to increase the size of the object. What do you think electronic microscopy they use? They use electron. Okay, means it is microscopy that use electrons to visualize the image, to see the image. So by definition, they use beam of accelerated electrons. Okay, they use electrons for illumination. Means to give the image, you require electron. Okay. Means you see how magnification it is much. So remember the magnification there. Magnification under light microscopy was not good, okay? Because was only 1,500, okay? Magnification under light microscope. But see how much electronic microscopy. It means it is 10 million. Compare, okay? That's why electronic microscopy can see even virus. Remember in the previous unity, we said that virus is seen only under electronic microscopy. To mean that by help of this electronic microscope, you can see very small, very small, because this light microscopy, this electronic microscopy, they have high magnification. They have high increasing the image power. Okay. I hope you are understanding. Means also their resolution. Okay, appreciate their resolution. Resolution, it is 1 nanometer. So I hope those who are doing the PCB, they are good than me to understanding those nanometer, micrometer, what and what. Okay, means also they have high resolution power than light microscopy. Very good. So all of this one which you are looking here, okay, all of these are application of electronic microscopy. First thing you have to keep in mind that application of microscopy is just to see virus. Remember. So in the previous unity, I teach you what you call kingdom monera. Kingdom monera, they have bacteria. Bacteria, it is among of microorganisms. When we say micro is small, microscopy. Okay, we use microscopy to see microorganisms. Remember the cell organelles. Under light microscopy, you are able to see mitochondria, rough endoplasmic reticulum, but under light microscopy, you can't. Okay, you are going to see large molecules like glucose, carbohydrate, proteins. Means those large molecules, you can see it. So under biopsy sample, means those people who are dead, when they are going to investigate the cause of death, are called the biopsy. So means under this electronic microscopy, you can see analyze the biopsy sample, and also you can see metals and the crystals. I hope you are understanding this, why this electronic microscopy is now made. It is very important because very small object, you can use this electronic microscopy to see it. I hope this is the image. I'm not going to teach you the parts of it, but even in your syllabus is going to be look like this. Just understand these things which is big like this. Okay, it's like a tank. Now they use what you call electrons. Okay, they use electrons. Now they see the image under high magnification. Okay, 10 million, and under high resolution. High resolution means they can increase much than electronic microscopy, and they can separate image. They can observe image separately. They have ability to distinguish 2 points very well. Okay. So coming on the advantage and the disadvantage of electronic microscopy. So when we say advantage, means they have high resolution power, they have high magnification, comparing to light microscopy. So means is able to visualize things we cannot see under light microscope. I hope you are understanding two functions, high resolution and high magnification. And then it is being able to visualize or to see the things you cannot see under light microscope. So when we see electronic microscopy are of two types. Means one are called transmission, other are scanning. Even their names are keyword. Okay, transmission means the electrons are being transmitted through the sample. while this one it's just only scan. Means suppose that this is the sample. Okay, if this is the sample, when we come here on the transmission, means electron will pass like this. Okay, while here on the scanning, okay, means they come and they scan. Okay, they scan. That's why they give 3-dimension image, while this one is 2-dimension. So I'm going to teach you this one in detail. So stay tuned, and then I hope you want to get the A grade in biology. And then remember that success is not accident. It requires to sit down. Once you sit down until now at this minute, it means that there is hope that you're going to get good grade in biology. So be courageous, stay positive. So among the disadvantage of the light microscopy, okay. So among the disadvantage, number one, it is expensive. So in Rwanda and even in Africa, maybe one or no countries which have this electronic microscopy. So means why we don't have this electronic microscopy, as we have seen that it is very, very important. It's because it is expensive. Not only to be expensive, also it sample preparation, elaborate sample preparation means to prepare the sample. Okay, sample preparation means sample to be analyzed. It is difficult to prepare. And also sample must be dry, cannot observing living specimen. Means once you need to observe living sample. Means suppose that you want to observe living bacteria. Means this cannot help. Okay? To observe the sample here, the sample should first of all to be dry. Means you should dry. Once you are dry, means there is no life. They are like a dust. Okay? Means they cannot observe living organism. So the image, it is typically black or white. It means the image of electronic microscopy never give you color. Okay, just through like just like a black or white. Okay. I hope you are understanding. So other things you have to keep in head to use this electronic microscope. It is difficult. It requires extensive training. Many trainings are required. Okay. Many training are required to be able. Means even me who are teaching this video, I don't know how to use electronic microscope. Means it is but light microscopy I know how to use by using coarse adjustment, those fine adjustment. Now move down objectives. Now your eyepiece and the objective, they contain lens. Now we are going to magnify by help of those mirror and what. Okay. So means they use high radiation exposure can damage your sample. Means some sample can be damaged by radiations. Okay. As I told you, two types are there, which all have high resolution, high magnification, but they have some differences. Okay. Number one, as I told you, it is transmission electronic microscopy. So I told you, means they study ITRA. When we say ITRA structure, are structure produced by electronic microscopy. So by transmitting, electrons through thin section. I told you, if you want to make like how can you understand these things? Means the sample is like this. They told you the transmission of electron through the section. If this one is your sample, as the name is transmitting, now the electron are going to transmit. Now you're going to get the image. I hope you are understanding the things. Okay. So let's continue by understanding the preparation. Means a specimen must be prepared, must be ultra thin. Means supposed to be very, very small. And in vacuum, means vacuum there is nothing there. Okay, you should prepare this. Means in vacuum there is nothing. That's why to observe the image of this electronic microscopy, its image, you cannot observe living organism. Means you should first of all dry it. After making it dry, as I told you, you should observe only observe non-living living organism. You cannot observe. Means if you want to observe bacteria, first of all, is going to be the diet. Okay, I hope you are understanding. Only keyword to put here. This one you observe image by transmitting electrons through the sample. While here you are using the electrons, okay, which scan the which scan the surface of coated specimen. You are seeing the difference. This one is transmitting the electrons, but this one they scan the image by scanning the surface of the specimen. Means they come and they scan the surface. That's why this one scanning electronic microscopy, they give a 3-dimension image. While a transmission electronic microscopy, they give it only 2-dimension image. The difference here, they scan the transmitting electronic microscopy. They transmit electrons through the sample, while this one they don't transmit electrons through the sample. But this one they scan the surface of the specimen. Okay. Now other difference. This one they give 3-dimension image, while this one is a 2-dimension image. I hope you are understanding those two types of electronic microscope. So what are home key to take away? Take away today on these types of electronic microscopy means both transmission and scanning electronic microscopy, they provide a valuable intercellular structure, okay, but each has unique capacity and limitation. As I told you, this one they transmit electrons through the sample. This one they scan the surface. This one they give 2-dimension, 3-dimension. What can you choose? I can choose this, which give you only. I can choose scanning, which will give me a good image, which is 3-dimension. Okay, it's a time to compare now. Right microscopy and electronic microscopy. In terms of the size, okay, means smaller, too light, easy to move. Means this light microscopy is small, as you are seeing here, but the image is not very. Even my magnification is not good, because this one is supposed to be big, but this one is supposed to be too small. Okay, means in the size, the light microscopy is small, while this one it is big, large. In terms of the cost, in Rwanda don't have electronic microscopy, means it is expensive, but we have many light microscopy there. Even in my school, secondary school, there was a light microscopy, means it is less expensive. Light microscopy, it is less expensive, while electronic microscopy, very expensive. Irradiation types means it use light. This one use electron. What about the control of the image formation? Means the image formation here, I told you we use lens. In light microscopy, there is lens, while this one they use what you call electromagnets. There is no lens in electronic microscopy. Okay. What about resolution? Lower. Right microscopy, there is low resolution, while this one higher, very expensive, higher resolution. Less expensive, low magnification. High expensive, high magnification. I hope you are understanding the difference between light microscopy and the electronic microscope. Image type. Usually, this is light microscopy. They give 2-dimension image. While this one, I told you there is 2 types. Transmission electronic microscopy, which give 2 image, and they transfer electrons along the sample. But this one, it have 3-dimension image, which is scanning electronic microscopy, as I told you. So by here, you are able to distinguish light microscopy from electronic microscopy in terms of size, cost, which light or electron they are using, resolution, magnification, or what and what. I hope take time and review. Take your pen and notebook and then draw this table from scratch. It is going to be fit in your mind. Remember, now NASA, it has changed the way of asking. They are asking multiple choice question. They can ask you, among the following microscopy, right microscopy, they have higher magnification than electronic microscopy, true or false? Now you get 0 if you don't read this, you don't listen this video accurately. So success is not accident, work hard. So by continuing comparison of light microscopy and electronic microscopy, so we are done in this one, this kind of repetition, we are here. Specimen thickness, okay, means usually the specimen, that sample to put in the microscopy on the stage. The sample should be thin specimen on the slide, but this one very thin sample to put in, means this one, the sample to put under here on the stage are thin, but this one under electronic microscopy, it is very, very thin. The color I told you, right, microscopy, you can get different colors, true colors. If it is blue, you will see as blue. If it's green, you can see as green. But this color here, it is only white and black. Means it is the first color electronic micrograph. Okay. Specimen preparation, it is simple, no much training. But this one very complicated, right? Electronic microscopy very complicated. Okay, harsh to prepare it and may introduce artifacts. So there is other things called living cells. I told you, right? Microscopy, you can observe living cells or living processes, but this one you can't observe living specimen due to vacuum requirements. Means remember, to prepare the specimen, you should put them in vacuum. Vacuum, there is no oxygen, no anything means organisms are going to die. Okay. Image formation, you all directly observe the image, but it requires what you call fluorescence screen or photographic plates. Okay. So limitation, it is living or dry specimen can be viewed, but living specimen cannot be viewed. Those are limitations. Means a disadvantage on electronic microscopy, you cannot see living specimen, but this one you can see living specimen. So the disadvantage of microscopy, raw magnification, raw resolution. Another disadvantage of electronic microscopy, there is no color. True color are not there, but under microscopy you can observe true color. So in terms of preparation of the slide, this one, I hope take time and review this table. After realizing this table, you will understand advantage of microscopy, disadvantage of electronic microscopy. How can you distinguish? They can even give you multiple choice to differentiate them, maybe in terms of color image. This one, microscopy, they give you true color, but here there is no true color, okay? Only white or black, as I have seen in the previous. So I'm ending my video, but let me have here what you call how to make the slide. See, this is the slide. If you are looking very well, this is paramecium, okay? This is paramecium, which is protista, okay? Remember, I teach you the kingdom protista. Paramecium is one of example of kingdom protista. Means under the microscopy, okay? Let me just have color. Under means this is microscopy, which is here, okay? This microscopy, these people, it is now observing. In his eyes, this is what he or she is looking in his eyes. Means they see things like this, okay? Means in his tools or any sample you put, if there is any organism, you're going to look at it like this, okay? Means there is what you call microscopic observation, okay? Means to observe microscopy, the objective is to observe a small organism like bacteria, amoeba, paramecium, okay? And the part of the microscopic organism like plant or animal cell or other thing. Means microscopic observation, the target is to increase the size of what I can't see, okay? Means if I want microscopy, my target is to observe the small object, either bacteria or what and what, or the part of the cells or what and what. So visibility, some details may not be unclear due to lack of color. Means once what you want to observe is transparent, means you can't see anything. Means if it is transparent, right, will pass, now you will see white, okay? Means to increase this time of visibility, I will teach you what you call staining, okay? Means due to lack of color, means those specimens which don't have the color, under microscopy you can't see anything, okay? Now how can you solve that problem? There is things what you call staining. So when you say staining, it's just to put the color on the object which don't have color, so that you can observe them under microscopy. Okay. Now other things is just distortion. Thin section can be distort or damage the structure. Means in the way you are cutting, for example, you need to observe maybe your skin or what and what. The time of the cutting, you can break down the if you want maybe to see epidermis of the skin, you can make your epidermis damaged. So there is another way to cut, what you call sectioning. It is the way we are going to make a section. When you say section are the parts, means we're going to take the our tissue or our target or our object we need to observe under light microscopy. There is what you call staining to give them color, and sectioning. Sectioning is to cut them well so that you are not going to damage the target you are going to observe under light microscope. I hope you are understanding. Let me teach you how the procedure are done. Okay. As I told you, we are having staining. Okay. What is the staining? It come here to give the color. See, this is one blue, this is one green. Means the staining, it will come and it will give the color what we need to observe. Means suppose that if it is transparent, you can't see anything. But by the staining, you are going to give the color to that object so that you can observe it under light microscopy. So other things, as I told you, is sectioning. Means suppose that you have the skin. You want to observe the element of the skin under light microscopy. Okay? So imagine this skin, you are cutting them to observe maybe epidermis or dermis. Suppose that now you can cut them wrongly if you don't follow the procedure I'm going to put here. Once you don't put the procedure I'm going to put here of the staining, nothing you will observe under your light microscope. So what is the function of the staining? It's just it give color. Means use color the stain to enhance visibility. Means without the stain you can't see anything. Means the function of the staining, it is to give the color so that they can increase visibility. You can see them accurately. Okay. For example, if you want to see the DNA, okay, I will teach you DNA replication and what in senior 5 and what all of them regarding to the DNA. Okay. You can use this stain called the gelatine violet to stain bacteria cell wall. So the cell wall of bacteria is called what? Chitin, cellulose, or what? No. Cell wall of bacteria is made of peptidoglycan. I hope you remember my previous video called Introduction to Classification, specifically in Kingdom Monera, specifically bacteria. Thank you so much. So in cutting, I told you that we are going to take our epidermis, okay, our skin, in things called wax. So that wax, when it is cold, we're going to now allow us to make section. So they are useful in the soft tissue like brain. Once you are going to have, you continue in university, and then you are going to reach maybe there is called what you call histology. Histology is the study of the tissue, where we will take a tissue and then we will do all of these things you are seeing, the staining and then sectioning. You will going to have much detail. So work hard. I hope those who are studying Candida Libre, they are enjoying the course, because there is no other YouTube channel you can see. You can see all this curriculum of NESA. Now stay tuned, work hard, make sure that you make subscribe, comment and share for those for your colleagues who don't know this channel. Thank you so much. So like doing this procedure you are looking here, okay. By doing sectioning and staining, you should be having some precautions. Otherwise, you're going to observe under microscopy maybe the things you put not in the sample. Suppose that you have stool, you need to observe if they have cholera. Okay. Now cholera agent, I told you it is Vibrio cholera. In previous video I teach you this is caused by bacteria, specifically cholera, which caused by Vibrio cholera. Suppose that here there is Vibrio cholera in this sample. Okay. The Vibrio cholera inside, while you are making sectioning and staining, you are introducing maybe Salmonella typhi, which cause dysentery. Okay. Now you're going to report to the patient that they are suffering from amoeba and dysentery, since it is your contamination. Here there are safety measures. You should wash your hand with soap, okay, with warm water after experiment. Use disinfectant to clean the surface where the bacteria may be present, and also be cautious for harmful substance and the organism. So I hope you are understanding how microscopy work. Apart from how microscopy work, you are understanding how to draw it. There are many parts. Okay. The types of them: light microscopy, electronic microscopy. I teach you that light microscopy can be either monocular or binocular, where you have two ocular or one ocular. I teach you how to make the difference between light microscopy and electronic microscopy. And then also I teach you the very, very important, the difference between magnification and resolution. How can you calculate magnification? Okay. Now, apart from that, I ended by how can you make slide preparation. Keeping header staining, very, very important in your exam, and sectioning. When they ask you what is the staining, is just to give the color to your sample so that you can increase visibility. What is sectioning is just cutting the sample, avoiding damaging it. So I hope I tried my best. This is making the end of my video today. Okay, if you enjoyed the video, please subscribe and then share to your colleague who are suffering to get a grade positive. So those students who are studying PCB, MCB, candidate specifically, sit down, study hard. Success is not accident. It is your Paul, the good man. I love you. Enjoy learning.